Okay, this meeting is officially being recorded. Thank you for coming to the first webinar. As we have explained a few times, this will be an opportunity for everyone to see how the work on the ontology is progressing. I ask all of those who are here today to bear with us as we ourselves are figuring out the process and figuring out what's the best way to get input, the best way to present things. So this webinar is a bit of a test trial. We're really trying to figure out what works best. We hope that as things progress, we'll be able to collect input in a more objective and formalized manner. But right now we're still learning as we go. I've presented this before, and we've talked about how our goal with seeking data interoperability is to improve data workflows with institutions, between institutions, or between domains, and when we report. And I think I didn't stress enough also interoperability with everything out in the world that is being done. There is a lot of, in the videos you should have watched prior to this first webinar, we've talked about data sets such as flights, for instance, that sites can crawl and fetch all over the world. And there are many initiatives to make data fully interoperable among systems. And that also happens in biology. So there is this OBO Foundry who is committed to providing interoperability of data in the biological domain. There's 158 active ontologies just on this field of biology ontologies. So there are many ontologies going on, and the initiative to make linked data, to make data interoperable, is much bigger than us. So why are we pursuing it? I mean, obviously it's state of art in data interoperability if there are so many ontologies, but of course we don't want to reinvent the wheel. Sure, the things that already exist and already have been created are actually put in practice. So we see a large number of ontologies and very few that have been put in practice or that have come all the way up to being implemented in tools. So we want to make sure that in the field of surveillance, ontologies are actually put in practice because it's such an interdisciplinary field. It's also about perfecting it, if not reinventing it. So if we think about some simple concepts of surveillance, for instance, you have a specimen, a sample that you collect, and then it was collected from some animal. It's going to be subjected to laboratory analysis to detect some pathogen. So all those concepts that you're seeing, there's already ontologies for them. There's already ontologies for pathogen classifications, for specimen laboratory analysis. This is just a list of some of those 158 ontologies on the OBO Foundry, which is Open Biomedical Ontologies, I should have said. This is just a list of some that may turn out to be useful for surveillance. The thing is that they model those specific concepts in their domain, let's say, and we see surveillance as this interdisciplinary field where this all has to come together. So we don't want to reinvent, we want to reuse, and that's the magic of ontologies. All the codes are reusable. We want to reuse it, but applying it to the context of surveillance. So how do we use those concepts and tag data with those concepts and make data understandable by a computer for the specific purpose of using it for population inference? We're working very closely with this genomic epidemiology ontology, which is the one that gets the active ontology that gets closest to our context of surveillance. The epidemiology ontology is unfortunately a deceased project. And also with this ontology of biomedical investigations, because it's structured for experiments, but investigations are not very different from what we do in surveillance. So if we want to join this world of open biomedical ontologies, if we want to make sure our ontology is compatible with all those other 158 ontologies, we have to comply with this structure, which is quite philosophical, but I kind of break it down here. So it's a top-level structure that tries to classify everything in the world into one of those categories. And we chose to do that. We chose to follow this path of being compatible with them by picking the things from this structure that makes sense for us. So we can keep everything that is in dashed lines here hidden. And our ontology starts considering that every concept we review can be classified as one of four things. It's a data item, it's an information, it's an actual material thing, a material entity, it's a quality of something else. So this usually goes for roles, like a veterinarian role, or a microorganism having a pathogen role. So this is a quality of some material thing, and a process. So this makes it more tackable. So we're just going to review all the concepts in our data and assume everything is either a material thing, a quality of something, a process, or an information or a data item about other things. So that's part of our process, is just try to classify our world of surveillance into one of those four categories. So as I said, as we progress through these webinars, we'll more and more come to a process, to a set process of how to review the ontology. On this first one, I'm just trying to introduce us to the idea that we'll review with you these steps that we do. So what are the steps? We get a data sample, and this here is a report we send. It's actually a small portion of SVA's annual report to AHSO. So we have one line for each of the surveillance activities we have to report for each of those pathogens. And then we have a number of already standardized columns that we report about our surveillance. So we're going to work like that. We're going to get a data sample, work with that data sample until we're done, and then move on to other data samples. So we're starting with data ultimately is that you could break this down into specific informations for each cell. But that's not how we think about data nowadays, and that's not how we collect data. So to keep the process, to keep compatibility with the way we enter data right now and we think about data, we're going to model the concepts, of course, column by column. So what does this column mean? What are the concepts we need to model the data that we put here, to have a computer checking that those data are compliant? And also, when we enter data, we enter it kind of row-wise. We think of our data as, each row here in this sample example is one surveillance system. So we are very used to enter data in this row format. So the first proposal we would like to make is that our ontology should be observation-centric, rather than trying to break up all the concepts into their unique cells, let's say. We want to keep a strong support to tabular data, for now at least, and recognize that in surveillance, we're going to have the most different types of data sets. It could be a data set with test results for each individual lab result. So each row is one lab result, or could be, like in the example I showed, each row is all the results of the surveillance of a whole year. So the observations we see for surveillance, they can have very different target populations or level of aggregation. But what they have in common is that they're information collected somewhere else, that we're trying to make sense of the observations in that data set for surveillance. So this is not as much of an objective question, but it's our first proposal. We're trying to get heads to nod and let us know if there is something intrinsically wrong about that, that our focus is not, for instance, on the animal or on the specimen that was collected. Our focus is on the observation. I have some observation here that can be used for surveillance, that has some health information about some level of my population, be it an animal, a herd, or an entire population of a country. And I want to interpret that observation to make use of it for surveillance.
Observation, about the word observation, is that for me, the word observation is more like raw data. And I think for surveillance, we use as well word from prediction, some information coming out of models that could be used for surveillance on another specific surveillance system. I don't know if you understand what I mean. It's just the word observation. I was thinking, for me in my head, it means raw data, but result from model could be used as well as an observation for other prediction. I don't know if you understand.
2 times I said for me observation means more, and then you said something and I couldn't get what you're saying. But I get your point, and I should have said that one of the requirements to be a noble ontology is that you don't name things names, but you actually name them something like this. So if you create a concept in your ontology, it would have something like this. Like it would have a code. So this observation wouldn't be called anything. It would be called, AHSO 000001. And then you create a label, a preferred label, and a synonym and an explanation. And that is specifically to avoid that people fight over the term itself. So it can have a preferred label, it can have an alternative label, the label can be in any language you want, and it has like synonyms and things like that. That's to say we shouldn't always just fight too much about the terms, but for observation, I actually wanted, I think I have a question later on this, I actually wanted us to discuss whether the word observation made sense or if it was too ambiguous. What we want to mean with this, what we want to mean is the record, each row in a data set, any data set that's going to be used for surveillance, whether it's a data set of laboratory analysis that each row would be a test result, or like a disease notification data set where each row is a new disease notification, or I don't know, a data movement report, each row is an animal died, or it could be, a report of surveillance where each row is a whole surveillance system and all the results of how many were positive and how many were negative. So could you write on the chat if you have any suggestion for how we could call that? How would you call that line of the Excel rather than an observation, like a record, or how would you call it? Could you write on the chat? This should have been a poll question. On the bottom right of your screen, we set a poll there, but for this first webinar, we couldn't come up with concrete enough questions to ask for polls, but maybe we'll come up with some suggestions. There are only 3 of you attending the webinar today, plus the 4 of us here. Some people have told me explicitly they will watch the recording afterwards. So maybe after we've given time for some people to watch the recording, we should circulate a poll asking what's the best term to use here. So if you have any suggestions afterwards, send them to my email. We're interested in any suggestions here because that's a very central concept for it's where we're going to start. So this is an important one. Thank you so much, Valerie. So here was the next input required, asking if the terminology was not too ambiguous, and then explaining that the unique identification is actually numerical, as I just showed on the chat. But we still kind of want a name that everyone can understand. I will show in a second, we reviewed other ontologies for that. Maybe I can just go on and see. Because step one is to find the concepts in our data sample. So we found these concepts. We decided we explicitly want to model each line in Excel, what that is. Not just the information that that line brings or the information in each column of that line. We specifically want to model what's the concept of one record or one observation, or however we're going to call it. So that's our step one, identify that we want to model that concept. Step two is review surveillance resources. This will be better exemplified next. Right now, we just know every surveillance resource has lines in a tabular format. And then the step three would be review other ontologies to see if this concept already exists and make sure if it exists, we reuse, because then we are immediately compatible with other ontologies. So we looked at these four ontologies here. So BFO is the one that's mandatory for us to comply with if we want to be compatible with all other biomedical ontologies. This is investigation artifact ontology, something like information artifact ontology. So it's the one that models information about other things. OBO is just different ontologies in that OBO foundry. Here's their name. Here are their names. And OBI is the one of biomedical investigations. And we found this concept of a data item here that we thought is where we want to go. And we reviewed actually everything that is in this data item, and we couldn't find something that represents quite what we want to say when we say this is one observation or one record for surveillance. So we think it will fit inside here. And this OBI, we don't need to go into this. This is like a Indian fight with arrows to every side. We don't need to go there. So we found this concept of a data item, and inside it a data set. And we think that's where our health observation should be. And we even think we should explicitly model when the results are aggregations of other observations, because for the data sample we showed where each line is a report of one surveillance system to AHSO, the way we got there was by aggregating, collating results from other data sets that were one result per line. So if we want to automatize some of the steps, we want the computer to understand the results when they are line by line, each observation, and when they're aggregated. So we think that's where we start. We start with the concept of a health observation that we may make a poll for the name. And we think it's a type of data item. We think that's the thing it is in the word. It's a type of data item. And then the step 4 would be modeling, which right now we mean modeling, we mean establishing the relationships between the new concept and every other concept in the ontology. Right now we only have one concept, so we can skip this step. But as I said, in future webinars, this is probably where we'll have more questions. So that's kind of something like that we would have to do. So as we add more concepts, we would want to understand how each new concept refers to everything else. This that you're seeing on the screen now is a very preliminary, rough draft of some of the concepts I think that were in that data set I showed you. So as we progress through the webinars, eventually we want to model this whole, that's how modeling a whole data set would kind of look like. And step 5 is repeat. So go back to the data sample, find a new concept, model again, and so on. It's a bit boring for the first one, but we can move on and go back to our data sample. And now that we have the concept of what a row is, we can see the first concept here is a zoonosis. Well, that's actually, what we're actually putting here is the microorganism that is causing that zoonosis. So it's actually, this column is actually listing microorganisms which are a type of hazard. So instead of thinking of putting the concept zoonosis directly on the ontology, we thought we should model that what we're actually looking for is what's the pathogen. But then we figured out that pathogen is just one type of hazard that we may be worried about in surveillance. And that's a bit going off script here, because the idea is that we should model the concepts in each data set, and then we start generalizing those concepts as we look into more data sets. But this is a bit of an inheritance we have now from RiskSur, where the concept of a pathogen, I kept thinking of surveillance the way RiskSur modeled it, and there it was a hazard. We specifically had discussions that things shouldn't be diseases, they should be hazard, because we could have things that are not diseases, for instance, toxins. So before even going into the concept of a zoonosis being some sort of organism that can cause a zoonosis, I went out there to look for whether I could find some resource for a classification of hazards. And I found this one that you're seeing on your screen that came from the project TAKIP that Matthias talked about in the meeting in Berlin. So if you know of any other resource that has a more explicit way to classify hazard, the different types of hazard that are not only microorganisms, if you in your data sets, in your institution, you've been using some type of hazard classification that is not this one, I would really appreciate if you could point me out to some resources. I reviewed some EFSA catalogs I will show in a second, however, the ones I reviewed. But I actually put the data catalogs for microorganisms inside here. And then I wanted some top level structure for how I classify hazards. And the only place I found was this TAKIP project. But either now on the chat or raising your hand, or later by email, if you want to point us to any specific way the data sets in your institution are being classified into hazard, please do so. And for those watching the recording also, they will be able to send to us by email. And just to show, because of course then the question is that concept already in the surveillance resources we have? We already have a lot of resources standardized the way we standardized in surveillance data, and in particular all the catalogs from EFSA. So I just wanted to point out that for all the concepts that I think are in that data set, we've already pulled out the specific catalogs from EFSA. So those are the catalogs I've associated with each of those concepts. And what we'll do through the next webinars, on this webinar, we're only addressing observation and hazard. But of course, in next webinars, we'll do more and more concepts in the same webinar. And what we intend to do is show you how we brought the whole catalog into the ontology, ask you things like, for instance, today, if it's okay that microorganism goes into a bigger structure of hazards, just presenting to you how we came from this catalog and how this became a concept in the ontology. And also, I'll be able to show you how we made sure that the ontology is fully mapped to the EFSA catalog. So the computer will actually be able to say, okay, this concept in the ontology is the same as the concept X in the catalog Y of EFSA. So we're making sure, so it's taking some time because we are mapping all these catalogs back to the ontology, and then we will present to you how those concepts became part of the ontology. For now, we started, as I said, from this TAKIP project that had some top classification for hazards. One of the types of hazards were microorganisms. And then from microorganisms on, we went with NCBI because, I mean, that's worldwide integration to everything people do. And what we did is actually within the Campylobacter species, Campylobacter genus in the NCBI taxonomy, we imported all the subspecies, all the species that the NCBI taxonomy has, and then we compared it to the EFSA catalog. So for instance, if Campylobacter avium is has a specific code a unique code in the EHSO catalogs, then in the ontology we said this is equivalent to the EHSO catalog, the EHSO term code, and then we put the unique term code from the EHSO catalog here. So what we have right now is a structure that is compatible with NCBI taxonomy, which is used worldwide, and mapped in full, so only for Campylobacter, I should say, because I mean, all the microorganisms is one of the hardest ones to tackle because it's so broad. So we really focus on where we're starting our pilot, and for Campylobacter, we've mapped in full the NCBI taxonomy to the EHSO catalogs. So I have another input to ask, in time, through the One Health pilots in Orion, we'll have a chance, the same way we're mapping now the EHSO catalogs, we'll have a chance to map to the ontology our own data sets, the data sets we use in our institutions. So right now we're mapping the ontology to the EHSO catalog, then we'll map our data set at SVA to the ontology, and then the ontology will serve as an automated translator between our data and EHSO standards. So we'll have a chance to, if we don't have any specific catalog of hazards at SVA, we just have the names that the laboratory people call the laboratory tests. And we don't have an actual catalog of agents, of microorganisms, we just have the names of the tests. So we'll in time have to map that, but there isn't a catalog in our institution somewhere of a microorganism list that I can just pull and make sure I can already put on the ontology. But if you do, if your institution use any catalog of hazards of microorganisms that is not the NCBI and it's not the EHSO catalog, I would also ask you to either point me to the resource right now on the chat, raising your hand, or think about it and send to me by email later. Again, if it's just not a formal catalog, if it's just the way your institution has been coding data, that will have to look individually at everyone's data. But if you use, I don't know, we use the ICAR classification or the ICD classification is common in the US. Some people use NOMAD. So if you adhere to a specific terminology or catalog of hazards or microorganisms, please make sure to point us to those resources. So here's where we are now. It looks like small progress, but took us a long time to map all those catalogs. So what we've done at the moment is we made sure to pull the catalogs from EHSO for each of those columns. And so far we've made them fully mapped or linked or translated into the NCBI taxonomy for Campylobacter. In time, what we intend to do is continue doing that. So mapping our surveillance resources to international resources. So for instance, here, the animal species, we can also map to the NCBI taxon. The matrix and the sample types, we've identified one very comprehensive anatomy or ontology of anatomy, and we also map the EHSO terms to that. So what we're doing is kind of going column by column, giving international interoperability, making sure we know how those terms map to those ontologies, so that immediately we can already make use of all the knowledge modeling that those ontologies. So right now we're going column by column, so we are making sure we give support to tabular data. So it's not moving away from the type of data we have right now, which I don't see as a bad thing, because if we have to support the way people are doing data right now, but we're trying to fully integrate with external resources. And even though right now we're still thinking column by column, as we start gathering and mapping the knowledge, we'll gain power to do semantics between columns. So we'll have, for instance, right now we have nice templates from EHSO to fill in those data that ensure that for each column we can only enter specific data, but it's very hard to check for consistency between columns. And that's something we'll be able to do once we start modeling how those concepts relates to every other concept. And I apologize that you can't really see this on this first webinar because we don't have enough concepts yet to start talking about modeling the relations between concepts, but we'll get there. As I said, our final goal is to map how things relate to each other and have the computer checking for the integrity of the data. So this is where we're going. As I said, we intend to map the individual data sources to the same key so that then the computer can do the job of translating data to AHSO or translating data back. So, you come with your institution data, we use the same key, then we can integrate it with our data here at SVA. So right now we're still focusing on so if you see AHSO as the output here, we're still focusing here. We're still doing this first step here. But rest assured, we'll get to mapping the individual data sources from surveillance that contribute to the surveillance data workflow. We'll get there. So we still have some time for discussion. If you have some open discussions that are not specifically the yellow boxes of input that we had, the next webinar will be in 5 weeks. For today, we just thank you for your attention. And if you have any comments or questions,